Our story begins with a monocyte that has differentiated into a macrophage. It then specializes and resides in tissue as tissue macrophage. When parasites attack, these cells act as the first line of defense, functioning as antigen-presenting cells. At this stage, components of the innate immune system, such as the complement system, are also activated. The ensuing cascade of reactions ultimately leads to the deposition of C3b fragments. These fragments later serve as tags, allowing phagocytic cells to recognize and target the pathogens. Other fragments like C5a and C3a play a key role in promoting vasodilation and increasing vascular permeability. Additionally, various chemokines are released, which are crucial for recruiting neutrophils and directing them towards the site of infection. Antigens on the bacterial membrane such as N-formyl methionine leucide, phenylalanine, along with products from damaged tissue like C5a and leukotriene B4 serve as important terminal chemotactic signals. Receiving these signals are neutrophils, which begin migrating toward the target in a sequence of well-defined steps, starting with rolling. Rolling appears as a loose interaction with the endothelium and eventually transitions into a phase known as adhesion. However, much has been learned beyond what traditional textbooks describe, so let's rewind and look again at the current understanding. The moving neutrophils initially get tethered to the endothelium through specific protein interactions. This may include the binding of P-selectin to its ligand, PSGL1, or interactions involving E-selectin and sialylated carbohydrate ligands. As rolling continues, these tethers are pulled to form a sling, which attaches to new sites on the endothelium and gradually peels away in a stepwise fashion. This process is followed by the engagement of cell adhesion molecules with integrins like LFA1. The interaction acts like a braking system, further slowing the neutrophil and leading to arrest. Also, during rolling, exposure to chemokine triggers conformational changes in integrins, enabling firm adhesion to the endothelial surface. This firm adhesion creates a stable base that facilitates the next step, crawling. Before we proceed further, I'd like to mention that these animations took a significant amount of effort to create. Over 1,200 keyframes were manually crafted. I made them available for free, and if you found them helpful, a simple gesture of appreciation, liking the video and subscribing would mean a lot. Let's take a moment to recap what we covered so far. Initially, tethers form and are stretched into slings. These slings then detach step by step. This is followed by arrest and then firm adhesion. What you're seeing here are intravital microscopy images that capture the actual process, how neutrophils tether, roll, and ultimately adhere. Once firmly attached, the neutrophil extends pseudopods and begin crawling along the endothelial surface in an effort to cross the endothelial barrier. Upon interaction with proteins such as PCAM1, the neutrophil initiates transmigration. It's important to note that in addition to the paracellular route, transcellular migration pathway also exists. Following the chemokine trail described earlier, the neutrophil moves toward the final target, where it detects opsonized particles. At this point, it has three possible mechanisms to combat the pathogen: phagocytosis, degranulation, and NETosis. In phagocytosis, upon recognizing the target ligand, the neutrophil undergoes actin reorganization and extends pseudopods that engulf the pathogen, forming a phagocyte. This is followed by fusion with granules, secondary and primary, leading to both non-oxidative and oxidative killing. Another mechanism is extracellular degranulation, where granules are released directly into the extracellular space. The release occurs in a specific order: tertiary granules first, followed by secondary granules, and finally primary, which contains the most potent antimicrobial agents. A third pathway discovered in 2004 is NETosis. This involves chromatin decondensation in the nucleus, followed by mixing with cytoplasmic and granular proteins. The resulting material is expelled from the cell, forming what is known as neutrophil extracellular trap. Nets serve to immobilize pathogens, enhance subsequent phagocytosis by neutrophils, and can also directly kill microbes via the toxic contents of granules and cytoplasmic proteins. The type we discussed so far is vital NETosis, in which the neutrophil retains functional abilities like chemotaxis, crawling, and even phagocytosis. There's also another type referred to as suicidal, lytic, or classical NETosis, in which the neutrophil undergoes a distinct form of programmed cell death that differs from apoptosis. As I now replay the entire animation, try to recall the various steps involved in this dynamic and vital process of neutrophil recruitment and action.